Hello, welcome to this PDC short video from the short video series for Transcend. Let's talk today about the Bradford Hill Criteria. I'm Dr. Mark Williamson. Let's get started. So a bit of a background. So the Bradford Hill Criteria were formulated by Sir Austin Bradford Hill, a British epidemiologist. And there are essentially 9 principles to evaluate the evidence of a causal relationship in epidemiology. So the list of them goes from strength, consistency, specificity, temporality, biological gradient, plausibility, coherence, experiment, and analogy. And then some have, after Bradford, have included 10th, which we'll at least talk about. It's called reversibility. So let's go through each of these criteria and see what they mean and how they relate to sort of causality in epidemiology. So first of all, strength. Strength, you could also think of this as effect size. And so the larger the association, the more likely the causality. However, caution is a small association might still be causal. It doesn't, if an effect size is small, it still may be real, it just might not be that strong. Like, for example, maybe, broccoli, causes lung cancer, but it's like, 1 in every million people. I mean, it might really increase the risk of lung cancer, just a tiny little effect. I mean, I don't really actually think broccoli does that, but this is an example. And so typically how you measure effect size is through something like for a t-test, it'd be Cohen's d. It's going to be a ratio of sort of the difference, like the mean difference between, like, say, a treatment and a control, and then the standard deviation. So signal over noise. And so a lot of times you calculate effect size for things like power analysis to actually detect, see how many samples you need to actually detect an effect if it's really there. And so effect sizes can be small, medium, large. And so the larger the effect size, again, the more likely there is to be causality between, say, something you see in the epidemiology, though not necessarily. But the greater the effect size lends more credibility. All right, moving on to consistency, aka like reproducibility. If you see this sort of cause and effect in multiple places. And so consistent finding in different settings strengthens the likelihood of effect. That's another way of saying it. So if you looked at a drug treatment on mice and you saw an increase in survival, and you saw that in a hospital setting, and you saw in the general population setting, they all sort of have the consistent type finding that really strengthens the fact that maybe there is a real effect here. It's pretty straightforward. Next is specificity. So what this means is the more specific an association between a factor and effect is, the more likely the causality. So the examples would be like, if you find a factor and effect in the a very specific population or site or for very specific diseases, the more specific it is. So say you have you're measuring some sort of virus or bacteria in a population, and you have these outcomes like hospitalizations. If you measure it there and you have a very specific association in this area, but you don't see that when you measure other areas that says this, there's more likely to be causality. Like I think one of the most famous epidemiological examples was like the cholera outbreak. I think it was John Snow, or who I think so. Anyways, they were able to like sort of trace the outbreak of cholera, the density of like cholera outbreaks to certain, I think, public wells or like public water in areas. And so they were able to say there's a very specific link between the actual potential source of the cholera and an actual disease manifestation. All right, moving on here, we have temporality. Again, for something to be causal, the effect must occur after the cause. However, just caution here. Let's see here. Yes, caution. There can be a delay in effect in play, so it might not occur immediately, but the effect should still come after the cause. So, for example, you have someone and they get sick, and you think that it's from like a rabid panda bite. Well, the disease, like rabies, like being hospitalized for rabies, has to occur like after the panda bite. It can't like happen before the panda bite. So temporality is an important criteria. All right, 5 is biological gradient, also known as dose-responsive relationship. So the idea is the greater exposure should lead to a greater incidence in the effect, both on sort of it could be on individual level. So like say lead, causes like health effects. If you're exposed to more lead, say in your water, you should get sicker. Or get at the population effect. So it could be, counties with greater lead in drinking water should have greater outcome, greater incidence of a certain disease that you're looking at. However, there is a caution: not all causes act this way. There's some things that might just cause something on just the mere exposure to it, and so adding more to it won't make it worse. So it's just like a 0 or 1. Maybe just even the smallest amount. Someone who's like really allergic, maybe the smallest amount of peanut will have a severe allergic reaction, where adding more won't really change. It will be severe either way. And so again, yeah, example could be, if you chocolate causing fever, more chocolate causing more fever. I mean, that's just a silly example, but again, it doesn't have to be this way for there to be a real effect, but typically a dose response is really strengthens the plausibility. Oh, speaking of plausibility, the presence of a plausible mechanism between a cause and effect increases the likelihood of causality. So if you have a mechanism on actually how this would work, really strengthens the reality of that being there. Now, this is caution. It is limited by no knowledge. Like there could be a real effect there that we don't know how it works. You might actually, maybe say, this radioactive gas causes brain tumors. You have no idea how the radioactive gas will get into the brain, but it really might cause that, even if we don't understand the mechanism, because we don't actually know that yet. Just because there isn't a known mechanism doesn't mean there isn't a mechanism. So like, again, for more of the plausibility, say you have someone's body, they get stung by a mosquito, which carries the disease, and then they get sick. So that's sort of the plausible mechanism of like a vector-borne disease. And so having a mechanism that makes it plausible for a cause and effect to sort of come together strengthens the case. All right, coherence. Coherence is so the idea is, if there's different types of findings, so if you have epidemiological findings, laboratory findings, etc., and they all sort of say the same thing, they cohere together, they come together, that increases the likelihood of an effect. Now, a caution here is the lack of one doesn't nullify another. But say you have a pesticide, you think it causes health problems, and you find that in the clinic, and you also find that sort of doing basic research in the lab, and that'll strengthen if you have multiple lines of evidence from different areas, sort of all converging on the same answer. That really strengthens your case. Finally, experiment, sort of dovetailing off of that, the more experimental evidence, the more likely the causality of the effect. Because remember, epidemiology tends to operate using observational methods, not really experimental methods. So look at populations and look at sort of possible like links between exposures and disease, etc. But if you can take those and actually go into the lab, do experiments on mice, cells, whatever, and you can build up a case there, that's going to have more evidence for it being sort of true in reality. And so, say microplastics in water causes weight gain. Well, if you have, a bunch of scientific studies that you actually show that in the lab, that's going to strengthen the evidence that it actually occurs sort of in the population. And finally, analogy. This is probably the most underrated one. Another way of saying this is stating similarities. So if you can use an analogy between an observed association and other known associations, can increase the likelihood of causality. So like, for example, for people, maybe that high blood pressure can lead to hypertension, and it's sort of like a silent, can be a silent killer. Well, maybe exposure to radiation like radon gas, can cause cancer and death. And so it's also a silent killer that builds up over time. And so the analogy is just like, high blood pressure is sort of hidden. You don't really have any warning signs and it can kill you in 20 years. So too is radon gas. It's not the strongest one, of the 9, but analogy can be useful. And then finally, reversibility, that's something that some people add sometimes. So the idea is the cause is removed, that effect should also be removed. Now, again, this is you should be cautioned. It could be very different on the individual versus population level. So, population level, this is a little clearer. So if you have lower an exposure to something in the population, and you think there's that is leading to some disease, it should lower the disease. However, on the individual level, that may not necessarily be the case, because say, for example, maybe you're exposed to something and even if you reduce the exposure, it sort of permanently damages you or something. So for again, an example of reversibility, say, exposure to pretzels, causes weight gain. If you take away the pretzels and the weight gain goes down, that seems to be help strengthen the case of the causality between pretzels and weight gain. All right, with that, we went through the Bradford Hill criteria from strength to analogy and then adding on reversibility. And this is useful for trying to go from epidemiology to not just observation, but sort of causality. And so hopefully this training was helpful. And with that, I will see you next time.